After studying this module, you shall be able to know about the principle and methodology followed for the northern and southern blotting procedure, learn about the applications for northern and southern blotting, and lastly, learn the similarities and differences between northern and southern blotting. A blot or the procedure of blotting is a technique that allows the transfer proteins DNA or RNA onto a carrier membrane such as a nitrocellulose or a PVDF membrane. This transfer is generally done after gel electrophoresis by transferring the molecules from the gel onto the desired blotting membrane, or can be done by directly adding the samples onto the membrane. Further, blotting is of different types depending upon the type of molecule transferred. For RNA, the name is northern. For DNA, the name is southern blotting, while for proteins, the name is western blotting. Northern blotting. Northern blotting is a procedure used to identify specific RNA molecules from a mixture of RNA molecules. It is generally used for the analysis of the RNA sample from a particular tissue or cell, which is used to quantify the RNA expression of particular genes. This process was invented by Alwyn and is known for its resemblance to the technique of southern blot, which operates over DNA. The common protocol followed for northern blotting is as follows. The initial step in any northern blotting experiment is to denature or separate the RNA within the sample into single strands. This step ensures that the strands are not bonded together through hydrogen bonds and are unfolded. The denatured RNA molecules are separated according to their molecular weight by using agarose gel electrophoresis. Following the electrophoretic separation, the resolved RNAs are transferred from the gel onto a blotting membrane. After the completion of transfer, blotting membrane carries RNA molecules in the form of bands which were present originally on the gel. In the subsequent step, the membrane is blocked and incubated with a DNA or RNA probe which has a sequence that is complementary to a particular RNA sequence in the sample and a molecular tag for detection of its location. This tag can be radioactively labeled molecule or any other tag that is non-radio labeled. After the probe has been hybridized or bound to a specific RNA fragment on the membrane, it can be detected among the different RNA molecules on the membrane. Applications of northern blotting. Northern blotting has multiple applications in molecular biology, biotechnology, and modern biology laboratories. The technique allows the scientists to observe the expression pattern of genes between multiple tissues, organs, developmental stages, and environment stress levels, pathogenic state of an infection, and also to monitor responsiveness to therapy. The technique has been applied to demonstrate overexpression of several oncogenes and downregulation of tumor suppressor genes in case of cancer as compared to the normal cells. it has also been applied to study the gene expression in case of rejection of a transplanted organ. Northern blotting can also be used to determine if the expression profile belongs to a previously known gene or when a novel gene sequence has been identified. Furthermore, the expression patterns derived on a Northern blot can provide insights into the gene functions if specific set of conditions have been used. Northern blotting can also provide information if a particular mRNA molecule undergoes alternative splicing. This can be observed if same probe is hybridized at multiple molecular weight levels. Alternately, if the product has lower molecular weight than expected, it may represent deletion or mutations in the gene. Southern blotting. Southern blotting is a laboratory technique used in the field of cell and molecular biology to identify specific DNA molecules from a combination of DNA molecules. Southern blotting is the transference of DNA fragments from an electrophoresis gel onto a membrane, subsequent in the immobilization of DNA fragments so that the membrane transports a semi-permanent reproduction of the banding arrangement of the gel. After the immobilization, the DNA can be subjected to hybridization analysis, enabling bands with sequence similarity to be attached to the labeled probe and thus is identified. Southern blotting merges transmission of electrophoresis separated DNA fragments to a sieve membrane, and the further fragment detection is done by probe hybridization. The technique was discovered by Edwin Southern and hence the name Southern blotting. Protocol for Southern blotting. The DNA to be analyzed is isolated from the source. Restriction enzymes are used to hydrolyze the larger DNA molecules into smaller fragments. The DNA fragments are then separated under the influence of electric field using agarose gel electrophoresis. This results in separation according to their molecular weight or size. In case DNA fragments above 10 kb are present, then the agarose gel may be treated with an acid such as dilute HCl prior to the blotting step. This acid treatment results in depurination of DNA fragments, breaking the DNA into smaller pieces, thereby increasing the efficiency of transfer from the gel to the nitrocellulose or nylon membrane. A sheet of nylon 6 by 6 or nitrocellulose membrane is placed on top in the direction of transfer from the gel. Pressure is distributed uniformly onto the gel to confirm a smooth contact among gel and membrane. Buffer transfer by capillary action also moves the DNA from the gel onto the membrane, where ionic interactions bind the DNA to the membrane due to the negative charge of the DNA and positive charge of the membrane. the membrane is overheated in an oven at 80 degrees Celsius for 2 hours, which are usual conditions for nitrocellulose or nylon membrane, or uncovered to ultraviolet radiation for nylon membrane to permanently join the transported DNA to the membrane. The membrane is then unprotected to a hybridization probe, which is a single DNA fragment with a precise arrangement whose occurrence in the target DNA is known. The probe DNA is labeled with radioactivity or a fluorescent tag for its detection. This probe can either be a DNA or an RNA molecule. In order to minimize the non-specific binding, salmon or calf thymus DNA is used for blocking of the membrane surface, and target DNA along with the deionized formamide and detergents like SDS are used to reduce non-specific binding of the probe. Following the hybridization, the extra probe is carried away from the membrane and the arrangement of hybridization is visible on the X-ray film by autoradiography or through development of colored bands on the membrane in case a chromogen has been used. Blots can then be probed using a radioactive or a non-radioactive DIG labeled probe. This probe binds to the target DNA fragment and helps in amplification and detection of such sequences. The hybridization reaction involves a buffer, a detergent, and a non-specific DNA to maximize the stringency. The membrane is washed with the buffer to minimize any non-specific binding. Detection. The blots are detected by autoradiography using the X-ray films placed along the membrane. Consequently, only those places in the membrane where the probe is hybridized with the DNA yields a band and thus can be easily detected. Hybridization of the probe to a specific DNA fragment on the filter membrane indicates that this fragment contains DNA sequence that is complementary to the probe. The transfer step of the DNA from the electrophoresis gel to a membrane permits easy binding of the labeled hybridization probe to the size fractioned DNA. It also allows for the fixation of the target probe hybrids required for analysis by autoradiography or other detection methods. Southern blots performed with restriction enzyme digested genomic DNA may be used to determine the number of sequences, for example, gene copies in a genome. A probe that hybridizes only to a single DNA segment that has not been cut by the restriction enzyme will produce a single band on a southern blot, whereas multiple bands will likely be observed when the probe hybridizes to several highly similar sequences, for example, those that may be the result of sequence duplication. Modification of the hybridization conditions, such as increasing the hybridization temperature or decreasing salt concentration increases the specificity and decreases the hybridization of the probe to sequences that are less than 100% similar. Applications of Southern blotting. Southern blotting is used to locate specific arrangements in restriction fragments that have been alienated by gel electrophoresis. This most noticeable use of Southern blotting is to detect DNA sequences that have analogous sequences to the one which are existing, that is, how many functionally associated genes have been known and considered. In this case, one strand of the gene being studied is applied as a probe. This information can also be applied to simplify a cloning experiment. For example, if the desired gene is exposed to be encrypted on a restriction fragment 6000 base pairs in length, then the fragments of that size can be separated from the rest and cloned. Further, it is also used to distinguish different strains of a given species by detecting subtle variations in their nucleotide sequences. Certain mutations create, whereas others put an end to certain restriction enzyme recognition sequences at particular sites in the genome. Therefore, when genomic DNA of diverse strains is processed with similar restriction enzymes, the individual DNA will offer to some extent a dissimilar variety of restriction fragment sizes. Each variation is called a restriction fragment length polymorphism, or in short RFLP. Southern blot hybridization is applied to selectively visualize restriction fragments that often vary in size. Similarities and differences between Northern and Southern blotting. While there are a number of similarities between the two techniques, there are significant differences as well. The most notable ones are on the basis of molecule detected. Northern blotting detects short-stranded mRNA, while Southern blotting detects double-stranded DNA. On the basis of gel electrophoresis, Northern blotting uses formaldehyde agarose gel, while Southern blotting utilizes agarose gel. On the basis of gel treatment, Northern blotting, since RNA molecules are single-stranded, the denaturation step is not required, while in Southern blotting, depurination, denaturation and neutralization is required. On the basis of membrane used, in Northern blotting, amino-benzyl oxymethyl membrane is used, while in Southern blotting, nitrocellulose or nylon membrane can be used. On the basis of blotting method, Northern blotting and Southern blotting both employ capillary transfer. On the basis of probes, in Northern blotting, cDNA can be radioactive or non-radioactive, while in Southern blotting, DNA can be radioactive or non-radioactive. On the basis of detection systems, in Northern blotting, autoradiography, chemiluminescent and calorimetric techniques are used, while in Southern blotting, autoradiography, chemiluminescent and calorimetric techniques are also used. In terms of use, Northern blotting is used to study expression of a particular gene, while Southern blotting is used to identify a particular gene. Northern blotting is a method applied to identify specific RNA molecules from a mixture of several RNA molecules. It is generally used for the analysis of the RNA sample from a particular tissue or cell and is used to quantify the expression of particular genes. The technique was invented by Alwyn and is known for its resemblance to the technique of Southern blot. It has multiple applications in modern biology, biotechnology and molecular biology laboratories. The technique allows the scientists to observe expression pattern of genes between multiple tissues, organs, developmental stages, environmental stress levels, pathogenic state of an infection and also to monitor responsiveness to therapy. The technique has been applied to demonstrate overexpression of several oncogenes and downregulation of tumor suppressor genes in case of cancer as compared to the normal cells. It has also been applied to study the gene expression in case of rejection of a transplanted organ. Southern blotting is used to detect the sequence of interest from the DNA from a variety of crude DNA sequences. It was invented by Edwin Southern. Southern blotting involves the transmission of DNA from agarose gels onto a nylon or nitrocellulose membrane. The membrane bound DNA is then stabilized and probed for a sequence of interest using radio-labeled or DIG-labeled probe. Most common applications of Southern blotting are in the field of identification of new genes, similar genes in different species, and in forensic science where it is used to support RFLP.